very much. While we wait for the slides, I got the honor to sit down, actually, not to have to stand. I don't know why. Is it because regulators take their time?
After lunch.
But all good. And yeah, some personal views here from an EMA perspective on how some of these projects could actually be useful also in regulatory terms. Does this work? Maybe you can move the slides for me? Oh, okay. So the ideas are grouped among the main themes of addressing the challenges of personalized medicine, making the most of observational studies and data sharing, developing new models of interactions more efficient in generating relevant data, and also something we've talked perhaps a little bit less this morning, frameworks for patient involvement into the benefit risk assessment. Next slide, please. We've seen a phenomenal list of challenges in terms of regulatory thinking. Perhaps what's most challenging is the consequence of segmentation of larger diseases into small populations and the difficulty of conducting randomized controlled trials. Now, it's not an insurmountable difficulty. In many cases of high unmet need and dramatic activity and a good understanding of the mechanism of action and safety profile, we've already been able to approve a number of drugs based on phase 2 studies. But as things get less clear-cut, it's going to become more difficult, and we're working on a concept of trying to identify a threshold of what could be a dramatic activity given all is known for a certain drug in a certain disease, and a threshold that we could decide in advance prospectively and look at as the single-arm trials recruit how we could do more clever external comparisons. Perhaps with all the knowledge that we have in terms of biomarkers, etc., this could result in a more efficient drug development in situations where randomized controlled trials are not feasible. Next one, please. Now, a lot of work goes on in terms of drug development, and one of the tricks will be to be able to bring them all together. So while we focused in the past on randomized controlled trials, in situations where this is not possible, well, there's got to be complementary evidence coming from a number of sources. And we've tended to be very skeptical about observational studies for the right reasons, because there are important issues on how one is able to manage bias. But again, perhaps if one could develop highly curated databases of observational data with much more information that perhaps could allow much better matching, perhaps we could reduce some of the uncertainty and manage some of these biases. And so methodological work in this area, I think, would be extremely useful. Next one, please. Endpoints are another favorite, a long story. We are still discussing validation of endpoints in a number of diseases. Well, this will continue as new modalities allow imaging endpoints to become more popular, all sorts of markers, and so on. How do we validate these important works that needs to be done? And I think will be has the potential to tremendously speed up development. The question is always one of methods and validity and clinical relevance in the end, because to do a benefit risk assessment, we need to understand a little bit, at least sufficiently, what it means in terms of clinical relevance. And there is also the opportunity to look back at other endpoints that perhaps we have neglected in the past, like at least at the regulatory stage of approval, health-related quality of life. If you think of it, there are today a number of tools available, perhaps too many even, and no well-agreed best practice methods on how a protocol for quality of life studies should look like, or what are the methods for dealing with the known problems of this data. And the more we use endpoints which are less close to something which is very clinically relevant and understandable, the more other endpoints like quality of life or other type of PROs will probably be useful in complementing the whatever imaging endpoints and tell us, hey, this is something that really means something for the patient. And it's not only for efficacy, but also for safety. Next one, please. Lastly, models of interaction. I think you all know that the way most trials are conducted today are, at least in Europe, is often the protocols can be developed without any type of input from academics or regulators, or at least not a significant one. And I think there it's where we need to break barriers and firewalls. Perhaps even us as regulators, we've been in the past too far away from this and only had sort of bilateral interaction with industry when industry wanted to have the interaction and so on. Whereas I think there we need to do much better and try to interact more systematically and earlier on. In drug developments, the way they're often done today, unfortunately, we're talking about personalized medicine, but the vast majority are very far from that ideal. Are big studies in unselected population. There is some type of positive benefit risk balance. The drug goes onto the market, and then all the additional work of identifying better the responders, the biomarkers, the dose, the duration, etc. All of that work is somehow not happening, at least not happening at the pace that it should. And perhaps there we need different models of interaction where perhaps academic centers take a bigger role in conducting that type of research and so on. It's all possible today. Doesn't need to be a massive innovation, but why is it not happening? Well, perhaps we don't have the right business models that make any of these very attractive for industry or for payers who would necessarily most likely want to have those type of data. So we need to think. ERTC has a fantastic platform for example, just screening patients with to identify very rare subsets. Well, these type of models exist. They're there. Why are they not used? Next one, please. In the last few years, we've done enormous progress in terms of data sharing, not just setting up some systems like Project DataSphere, which I think is extremely successful, but also shifting the mentality from a time where people thought this was counterproductive, stifling innovation, and not feasible for a variety of legal and other impediments, to a situation where these platforms exist, and even the most skeptics, I think, understand that this is a tremendous potential to foster innovation. So we need to move forward in that direction, not just in sharing high-quality databases, but also looking at the post-marketing phase and all the electronic health records to start answering some of those questions that often at the time of approval are not yet possible to answer. And also facilitate long-term follow-up data collection for clinical trials, which is often missing at the time of approval. Next one, please. And lastly, this is what we've talked a little bit less perhaps this morning, is the concept of benefit risk analysis. It's a critical concept for regulators, but today it's done still largely intuitively. Do we know exactly what patients think in terms of how much they value certain effects of drugs, how those preferences change over time, and so on, so that this data can be available for regulators when making decisions, but also for patients when making their decisions, and so on. And also, everybody has now, well, many organizations have come up with their own value scale, but these are similarly, you know, a group of experts saying whether, I don't know, a hazard ratio of 0.8 is clinically relevant or not. Well, what do the patients really think? Do we have evidence from patient preference studies of what they think and how the different drugs rank between each other based on evidence? Next one, please. Lastly, and on this I want to finish, but it's actually a very important point. How do we as regulators interact with all of these programs? We are, as I said, happy to be in communication and advising from the very beginning, from the ideas to the implementation and so on. On occasion, we will also be part of some of the consortia or take a more active role once a consortium has won a call and has been adjudicated a certain call. But what is more important is that as regulators, we have a number of tools like qualification of methodologies. And it's very important that any of these programs, if they have any type of regulatory step in the end, or that they go through those processes to make sure that these are not just academic exercises or industry-driven explorations, but that when these new methods come to the regulatory stage, that they actually are acceptable. And for these, it's not just a matter of collaboration, but it's a matter of using the existing qualification tools that we have, be it drug regulators or devices or whatever. Thank you very much.
Thank you. Any questions? I have one. I mean, we talked in the morning a little bit about a kind of a virtual control arm in order not to have, take the example basically what David put forward also from the PD-1 or PD-L1 studies. So, I mean, there's 5, 6, 7, 8 companies developing these antibodies and the checkpoint inhibitors. So the question is, everyone runs similar control arms, so to say, standard of care. What do you think from a regulator point, what would be needed in order to kind of skip it?
So it's a difficult one, because today, if we ask our statisticians, they will tell you that no matter how good your whatever propensity scoring or matching algorithms are, you're never going to get away with a control arm that is comparable to a parallel control. Mm-hmm. So there are, for me, two ways forwards. One, okay, the methods are imperfect, but how much uncertainty is there still left? Okay, to make very minute comparisons, probably this is not suitable, but if you have relatively big effect sizes, even if not dramatic, perhaps you can draw some conclusion in that situation. But much more interestingly would be the approach which, like the LangMap protocol, where you actually have concurrent experimental arms sharing one control. So that level of cooperation is difficult, I know in practical terms, but I think that's the other solution to explore before actually. My personal view is that if the data sets that we use for external control are actually much more comprehensive in terms of data collection, then we must be able to reduce the level of bias. So if perhaps in certain diseases we understand so much about the prognostic factors that we are able to do to decrease the potential for bias, I think that should be feasible, but still to be tested.
Okay, thank you. Andreas has a question here in the front, please.
Yes, thank you. I'm Andreas Voss from Caris Life Sciences. I'm wondering what does it take to move away from the concept of controlled trials in this day and age? We have data that is accumulating that says that there are no two identical patients. So the concept of a controlled trial is based on comparability between the patients. And I think that comparability is more and more in question now with the data that we generate about the genomic diversity between patients, and I think it is soon obsolete to speak about a control arm. How do you see that?
Yeah, I understand very much the philosophical discussion, and it's true when patients are randomized into randomized controlled trials, you don't know at all what would happen to a patient if he had taken the other arm. Could have been much better, so it's really not a tool to tell us which patient does best. It tells us only on a population level what are the results, but it's a difficult one. In terms of generating evidence, it's still considered to be the golden standard, But for me, there are a number of situations where you can draw conclusions on even externally controlled trials. And our wish is that enough methodology is produced and validated to tell us, look, you can use these methods to decrease the uncertainty from this to this. And then there is a whole set of effect sizes that you can perhaps confidently show.
Olga?
Within our strategic governance group, Oncology, we also started discussions about combos of various drugs, and we touched upon this already this morning. And so we may come at one point in time also to a combo of drugs which are not approved for certain indications, which is, by the way, often also a matter of debate within the companies. And so what's the view of the EMA on this? And we have seen the examples this morning that certain mutations may come up in other indications where the certain drug is not approved and so on and so forth.
From a theoretical perspective, it's not at all excluded that one could approve, let's say, a set of drugs, each of them not approved and only working together, so-called novel combinations. If it's shown that it works and it doesn't work outside the combination, or at least you have data to tell you that don't even try it, not in combination, why not? It's not a problem. I think we would probably struggle a bit to find the correct sort of regulatory framework to do this, but I don't see it at all impossible. If that's the way forward, we still have to see it, but that even algorithms, in theory, algorithm of assigning certain combinations could be part of a regulatory label. That's not at all excluded. If it makes sense. In this sense, regulators will just probably follow the science. Today, I don't think we're there yet.